All right, welcome everyone to our August 26th Rutgers Ornamental IPM webinar. We're going to be talking about some common issues that we're seeing out in the nursery and the landscape this time of the year. My name is Bill Erickson. I'm the agriculture agent for Rutgers Cooperative Extension of Monmouth County. I'm also joined by Steve Retke, our IPM program associate, Tim Waller, our ag agent in Cumberland County, and Aaron Quinn, our IPM program support staff. And so just a reminder on the disclaimer on the bottom, we're going to be talking about different materials and chemicals to control different insect and disease issues. So just make sure you always follow the label. And if you're in a state besides New Jersey, your regulations may be different. And if you're a landscaper versus a commercial nursery grower, those guidelines and recommendations may also be different. So always follow the law and the label on the material. Okay, so today we've got a good lineup of different issues that we've been seeing. We're going to talk about some beneficial insects, which are dragonflies. We'll talk about leafcutter bees, a new insect that we're seeing more of, the flea hopper, nostoc, nickel deficiency in river birch, some of our tulip tree and magnolia scales, the Douglas fir needle midge, slugs and snails, banded ash clearwing moth borer, and verticillium wilt. And if you look on the bottom of the screen, you'll see our current growing degree days. In North Jersey, we're around 2,500. Central New Jersey is 2720, and Southern New Jersey is up around 2875. So those are the thermal units that have been accumulated thus far, and they're actually pretty close in range for the three different regions of the state right now, getting towards the end of the season. And you'll see that on each slide as we progress through, just to give you a reminder of where we're at and where our different insects all correlate to those growing degree days. A couple of updates before we get into it. There's a new app out there that's really useful from the Resistance Action Committee. And so we're able to look at our different groups of fungicides, insecticides, and herbicides all in one place. So you don't have to look on different apps or different websites where these used to be split apart in different areas before. Now you can download this one app and they're all in one place. And this would be if you had to look up like the group number, for example, for your fungicides, so that you can rotate your chemistries. And those group numbers are based on mode of action, and we rotate them so we don't develop resistance. This app can help you to identify what group each of your materials are in. And so there's a lot of really good information on this app. So this is super helpful tool that we have for IPM. Another update is that we're starting to see some second generations of a couple of pests that we've already talked about that had their first generation earlier in the season. Japanese maple scale and red-headed pine sawfly were both in the window of their second reemergent generation for this season. And so we talked about those already. The QR codes will take you to the spot on our website where we have posted the information about them, or the links on these pages will also take you there as well. So when we send out the digital PDF versions to everyone, those links will be live and you can just click on them. Or if you have a printed version, you can scan that QR code with your phone and it'll take you right there, and you'll have the information on the life cycle and controls of these insects. Same thing with our website. That link will be live in the PDF version, and the QR code will take you there. And on the website, if you haven't been exploring it recently, we've been putting all of our videos on there, all the recordings from the webinars, and then we also have an insect and disease database that we're building with all the information, so you can click on each of these pests and find the information. It's all available there on the website. Okay, so let's get into it. The first insect we're going to look at are dragonflies, something that a lot of folks are familiar with. They're really beautiful insects, and these can actually be really beneficial for us as well in the nursery or in the landscape. They are flying insects, as we know, and the larger species can fly up to 30 miles per hour. So they're really agile in the air. They can be really quick, and this helps them catch their prey mid-air. So they have a 95% success rate. Their eyes can see 360 degrees practically around their head, and so they're really good at catching aerial prey. And they'll feed on mosquitoes, flies, butterflies, moths, bees, wasps, anything that flies and that is small enough for them to eat is fair game. And so these can really help with keeping insect levels down in your operation. There's two different types of dragonflies. Well, there's many different species, and there's two different general classifications. The percher types are going to make frequent stops and perch on an object and kind of wait there for their prey. And then patroller types are in constant motion with a larger range, and they're kind of always moving around. So one of the things that's really interesting with dragonflies is that we're usually more familiar with the adults, is what we're looking at here. But the nymphs, which are also called naiads, those are actually aquatic organisms. So the adults are going to lay their eggs in mud or in water. They need a water source. And then the nymphs hatch out, and they can actually live in that nymphal stage for up to 2 years. So a large part of their life cycle is actually as an aquatic organism. And then eventually they're going to grow and turn into an adult, and the adults only live 2 to 3 months. So the life cycle of the dragonfly that we're most familiar with is not even the longest phase of their life. So it's really interesting that their life cycle can either be 1 year or can be several years long if they're actually spending that long underwater. So we can see the adult, which we're familiar with, really small antenna, the eyes that can see, almost around 360 degrees, long, narrow abdomen, and then of course they're characteristic for large, clear wings. But on the right there's the naiad. That's the aquatic form, the nymph. And so this is something that we usually don't see because it's living in the water, but it's represents a major portion of their life cycle. So if we want to encourage dragonfly populations at a farm or on a landscape, having a permanent water source is important, right? So they can lay their eggs and that nymphal stage can exist. Having diverse vegetation, aquatic and terrestrial plants to bring in the prey for them that they can consume in both of their life cycle stages is important. If we're talking about the perchers, we can have perches and sunny spots for resting and hunting and keeping some leaf litter there. And it's just another plug for avoiding, broad use, avoiding the use of broad spectrum insecticides whenever we can, and kind of trying to spray only when necessary, targeted applications specific for the pests that we're trying to control, reduced risk materials, all of these things can help to foster our beneficial populations. And, maybe sometimes you have to use broad spectrum materials because you've got a major outbreak and that's the only material that's really effective. And, it's just balancing that as part of a broader IPM program to reduce those impacts. And so leafcutter bees kind of bring that same point up in an interesting way also. So we start to see these late in the spring and persisting throughout the summer, and we're starting to see what we're looking at here. This is cercis, this is a red bud leaf, and you see those distinct kind of circular chew marks on the margins of the leaves. And this is a symptom of leaf cutter bees. So we see this, in the nursery and in the landscape. They can feed on many different species, flowers and leaves as well, but redbud seems to be, one that they really prefer. And so these are really interesting organisms. They're about the size of a honeybee. They kind of look like honeybees, but they're a little bit darker. They're almost black, and then they have these white bands on their abdomen, and I'll show that to you. So what they do is that they construct their nests in narrow cavities. So if you've seen like bee boxes or bee houses or things like that, this is one type of bee that can use those to construct their nests. And what they're doing with the leaves here is that they're cutting these pieces out of the leaves and using it to build cells within that nest area. And then so they build a cell and they fill it with pollen and nectar as a food source, and then they lay their egg, and then they cap the cell over, and then they build another one. And so this is how they create their nests and how they reproduce. So in the landscape, maybe it's not as much of an issue, and you can talk with your clients about, oh, this is, you have leafcutter bees. That's what this is evidence of. They're not going to destroy the whole plant or damage too much, but it could be a challenge sometimes in the nursery if you're trying to sell plants and they're showing this damage. So interesting species, and the young, once they hatch out, they're actually going to chew their way out of that nest of leaves and pollen and nectar, and then they emerge and eventually become adults. So this is what they look like, and you can see they've got those mandibles on the front, on the close-up picture on the left, the jaws, and that's what they use for cutting the leaves. And then on the bottom, we can see the cells that are created, that they're actually building using this leaf material, then filling it with the pollen and nectar. And they do look like honeybees. From afar, if you're not familiar with one versus the other, you may confuse them with the honeybee. But upon closer examination, you can see those white bands on the dark abdomen, and that's going to be characteristic of the leafcutter bees. So we generally want to avoid spraying whenever possible but there are other things that we can do to try to limit their damage in our operations. So these are actually really good pollinators, especially for some agricultural crops like apples and legumes, alfalfa, but they have an important ecological role. And so we may want to limit their populations from damaging our crops and our plants, our ornamentals, but there's ways that we can do that culturally without really having to spray them. So there are several natural predators that will actually keep their populations in check in nature. And then there is another species of leafcutter bee which will lay their eggs in the nests of other leafcutter bees, and then they eat the young of the bees. And that's kind of the ecological food chain happening there. So they can actually be predators of one another. So they don't sting people. They're not really territorial or don't really guard their nests as well. So they're not a danger to humans. Things we can do, we can cover plants with netting or use like shade houses that are enclosed in some situations, especially if we know certain crops are susceptible. They will try to create their nests sometimes in the pruned ends of roses. Like when roses are getting pruned, they may try to bore out that cavity. And so we can cap that up with thumbtacks or glue or like wax those ends that can keep them out of there. If we look around for holes in wood that might be used for a nest, those hollow cavities, that's what they're going to look for. So try to reduce that, try to fill that up. If there's any decaying wood around, remove that as well. So we're trying to just reduce their habitat, make it less appealing in the environment where we don't want them to be if we want to discourage them. So this is a fairly, I don't want to say a new insect, but we're starting to see it more and more in some of our nurseries, and some of our cooperators have shared some good information on this insect with us. I know Steve Redke has been seeing it when he's been going out and doing scouting, and this is the flea hopper. And so there's some information out about it, but we're trying to compile that and share it, and also to keep an eye out for it to see if it's showing up in your operation as well, and to let us know so we can get a better handle on this. And so they've been first observed back in May, and they're still persisting. We're starting to see that damage. There's a wide range of plants, and I'll show you some of the ones that we've seen that have been a little bit more susceptible than others, and then also weeds and grasses. And that's an important thing to remember. We'll talk about that on cultural controls. And so this is what we're looking at here. They're about the size of fleas and they jump. And that's why they get the name flea hopper. And so they're small plant bugs. So that means they have piercing and sucking mouth parts. And we'll see the difference. There's a sexual dimorphism where the females look different than the males. We're looking at a picture of the male right here, and you can see those white spots on the male. And so that might catch your eye on a plant when you're scouting, more so than the female. So I'll show you some examples of them compared to one another. They're going to lay their eggs sometimes in the stems or leaves, just so the tip of the egg is in the surface. And that's kind of significant because it may take repeated applications because of where the eggs are laid. We'll show you pictures of the newly hatched nymphs that are pale green. They almost resemble aphids to some extent, so you may confuse those as well. And the adults can live from 1 to 3 months, which is kind of a long time for an insect like this, and there can be 5 or more generations per year. So they can be pretty persistent throughout the season. We'll see some of the symptoms. They make these pale spots on the upper leaf surface, and then the signs, we'll see excrement and frass on the lower surface. And so this can cause leaves to become chlorotic, turn yellow, and drop off prematurely. And so this is what they look like, and some of the damage you can see on the bottom, 2 bottom photos showing the damage. Again, they have that piercing and sucking mouth part, and they're going to cause that stippling damage on the leaves. See the female on the left, so she's a little bit more, has a rounded shape, where on the right, the male is more elongated, has the white spots, or the female is just about fully black. And in the middle, that's the nymph that start out in that green phase. And if you just saw that quickly, you might think it's an aphid, but it's a different insect. This is the flea hopper. So this is some reports from one of our nursery cooperators who had been seeing some flea hopper damage, and they shared this with us. And we really appreciate this, and we can share it with all of you, is looking at which of their plants seem to be more susceptible to the flea hopper damage. And Buddleia and Salvia seem to be ranking highest on the list. So if you grow either of those plants and you're not sure if you have flea hopper or not, you may want to go and check those, scout those first, see if you see some of those symptoms, that stippling damage, and then take your hand lens and see if you can find them on the underside of the leaves. Monarda, Agastache, Phlox, Nepeta, Helenium, Veronica, those are all potentially a little bit high, medium to high susceptibility. And then we've got some plants that are showing medium to low susceptibility as well. But I would start with, if you're growing any of those high or medium to high susceptible plants, go out there and scout that out. And you can reach out to us if you're not sure, or if you want to get more information about it. But this is something that we're just starting to see more of it and getting more reports from different nurseries. So here we can see the symptoms a little bit closer. This is Buddleia, seems to be one of their really preferred hosts. You see that stippling on the top of the side of the leaf, that little bit of discoloration, and that could present like many different symptoms of insect damage or abiotic stress. But then you turn it over and you can see the signs of the insects. So some of that frass on the bottom, you may see the insects themselves. Take your hand lens and look at that. There are some natural predators that will help with this control. And then another really important cultural practice is to remove weeds in the area. So we mentioned that weeds and grasses can be alternate hosts for the flea hopper. So I want to keep the areas clean, keep the weedy areas to a minimum. Rotating your crops, even if it's a container nursery, if you had something that was susceptible to flea hoppers, grow it in a different house or in a different part of the nursery. So we can practice crop rotation even in the container nursery, putting covers on susceptible crops or putting them in a screen house, disposing of debris and waste so there's no extra food source. There's several materials that are listed for control, are pyrethroids, carbamates, and spinosins on the biorationals. But we've been hearing that the acephate seems to be the most effective. So again, that's some of what our collaborators have been sharing with us, is that they think that really seems to be the most effective treatment. So if you have other experiences also, anything with this insect, definitely let us know, and we can talk more about it and share that information with other folks in the program. All right, I'm going to turn this over now to our middleman, Steve Redkey.
Okay. Hello, Bill. Thank you. And hello, everyone. Good afternoon. And yes, I am the middleman of this trio of presenters. And today I'm going to speak on nostoc, nickel deficiency of river birch, magnolia and tulip tree soft scales, and then also the Douglas fir needle midge. All right, let's go and first with nostoc. Nostoc is something that, even though it's been around for decades, I personally was not all that familiar with it until maybe about 15 years ago. I'm kind of a slow learner. I should have learned it before then. But it's a cyanobacteria, And for years, it was mistaken as a blue-green algae, but it's not that. It's really a primitive growth, and it's an ancient organism. It's apparently 3.5 billion years, and so it's really quite ancient. It doesn't have any kind of, it's not a plant, it doesn't have any roots or vascular tissues, but it is photosynthetic. And what's interesting about this is that it poses a real slipping hazard. It's not a problem to the crops. It's not toxic. In fact, apparently you can even consume it. I certainly would not do that, but that's possible if you want to be part of your diet. But they have this gelatinous masses composed of filaments that's covered with a jelly-like covering that makes it extremely slippery. I always kind of relate to the fact that if you ever walked on wet ice, I mean, if you're walking on wet ice, unless you have spikes on, you're going to be falling. And so that's something I want to avoid when I go into nurseries. All right, there's a couple close-ups of them. They do form initially these types of bubbles, these filament bubbles up. You can distinguish that for, say, from example, from a moss or other algae, which don't have that. They're more of a smoother type of a texture to it, whereas this nostoc has this bubbling three-dimensional look to it. And then at the bottom, you see these masses of gelatinous filaments. It's really quite interesting to look at. And where I see the most, of course, working in nurseries is on ground cloth within the nurseries. And so when I walk into a container bay, which, these container bays have prolonged irrigation, sometimes 2.5+ hours every day in some cases, that's really an ideal environment for the growth of nostoc. And so if I walk into the nursery and I'm knowingly not paying attention, I could easily slip and fall. I've never done it, but I've talked to some of the various nursery personnel at nurseries where I work with, and they say they have seen people fall, and usually they laugh until someone gets injured, and so it's not so funny anymore. But they are, and as I already indicate, they're not toxic, but they can potentially, not a problem for plants, but they can potentially harbor fungus gnats and snails. And it's showing a series of photographs. And I'm going to go kind of a clockwise motion, starting at the top left. You see moss example. Obviously, that is not a slipping hazard, and it's really quite easily to distinguish moss from nostoc. And then you walk into a container bay, as shown there in the upper center, and that is a real concern. And I'm not going to walk in there probably. If I am, I'll be very ginger. And then you can see as we move further clockwise that really can be quite a mass of these filaments with this protective jelly type coating. And actually, when you want to control this, which there are chemical controls, herbicides that are available, the ones on the far right, the upper and the lower, those are ideal situations where the nostoc is nice and moist and is hydrated. And that's when you can get your best post-emergent control. And I'll talk about that a little bit later. And then as we move further at the bottom center, you see that they're kind of changed coloration and they're beginning to dry out and desiccate. And then finally at the far bottom left, you have a desiccated former mass of nostoc. Now, that's not dead necessarily. Nostoc can tolerate a fairly long period of time being desiccated before it dies. And so all it needs to be is rehydrated again, and it forms back quite readily. All right, as far as management of this, so you can certainly do mechanical raking. There's a little bit of labor involved, blowers. You try to improve your drainage, and there's a few other novel type approaches that maybe sometimes a bit on the expensive side. But you do have some of these materials that have shown various levels of success, things like the coppers and the ammonium chlorides and those types of things. They have been somewhat inconsistent with their controls. Sometimes they can be quite good. Other times they're maybe fair at best to trying to control and reduce this nostoc. Only 2 years ago, research done at the North Carolina State University by Dr. Joe O'Neill, the plant pathologist, rather the weed specialist there, he determined with research, with confirmed data, that glufosinate, which is the brand name Finale, provided just outstanding control. It's the best post-emergent herbicide available to control nostoc. And I'm not going to have time to go over the various types of treatments, and they have different concentrations. Usually just one or two applications is all that's required. And you can get 90-95% control very consistently as well. But just to emphasize under the considerations that nostoc must be hydrated for post controls to work, because if it's dry, it will be ineffective. All right, now going off to another kind of interesting thing, another one that I kind of was first fuddled by a good decade, maybe it's been about a decade now since I first saw this at a nursery, and I've seen it at a couple other nurseries since then. And these are with river birch, Betula nigra, containers too. Not so much ever going to be probably seen in mineral soils in the landscape. You're just not going to see this. It'd be quite rare. But in containers, it's a bit more common. And what's happening here with nickel deficiency is that it creates a stunted growth with very short internodes between the branches and very small mouse ear type sized leaves. And it's often been called the mouse ear disorder. But you can see the difference in the sizes, and those smaller stunted plants, you're not going to be able to sell those as a nursery grower. And so you can notice the ones that are full size versus the smaller ones. All right, so the host plant is really primarily river birch, and that's really the only one I've seen it, the Betula nigra in nurseries, although apparently it can affect a couple other, 2 or 3 other plants potentially. But it's been determined that when pH gets higher than 6.8 in nursery soilless media, now we're talking containers only, that the river birch is very inefficient at absorbing some of the heavy metals, especially things like nickel. And that's when these witch's broom symptoms can occur. They seem to occur just at the upper, newer growth primarily. The lower growth may be normal size, the leaves. And so apparently this nickel is it's not apparent. It's not a mobile nutrient. And so it can't move from the lower leaves to the upper leaves. And so you get that effect on the upper growth primarily. And so, yeah, it just indicates some of the symptoms are shown there. You've already seen the photographs, and the dwarf foliage, the blunt leaf tips, and the brittle shoots and so forth, the leaf chlorosis. And as I already indicated, nickel deficiencies rarely occur in mineral soils. In fact, interesting as well is that if you take a container plant with this nickel deficiency symptom and you place it in the landscape, it'll recover. The nickel will then be able to be taken up, probably, and the leaves will turn normal maybe within 6 weeks. It'll look perfectly fine again. All right, just another couple quick photographs showing you the stunting of the leaves, that mouse ear type symptoms, really tiny, just a small fraction compared to our normal size leaves. And it's going to be at the upper foliage. And now this is kind of more technical stuff, and some infancy new stuff that our staff support came up with. The fact that what's really the cause of this is maybe a number of different reasons, but apparently the reason the plant can't take up the nutrient nickel so much is also it has an excess amount of urea that's causing acid buildup to occur in the plant, even though the soil pH is high. And so that causes apparently a conflict, and the plant can't take up the heavy metal nickel. All right, and also some fertilizers as well that can exacerbate the problem. So you have to be aware of that. And some maybe unusual things like root knot nematodes or very cool soils can create these types of witch's brooms as well. But the key thing is, and what was discovered at the nursery that initially found this is that, and with research from other universities, that when pH gets below 6.8, the chances of this occurring becomes much reduced. So when you get pH up to 6, 9, 7 or more, then the chance for, in container river birches, the chance for this is becoming a higher potential. Okay, and you can apply nickel sulfate. This is where it was quite effective as either a foliar or a drench application. It doesn't take much, like 500 parts per million, or 5,000 parts, or rather 5,000 pounds per cubic square yard. So just a little dab will do you with this, and you don't want to over apply because you create phytotoxicity with the excess amount of nickel sulfate. And it's again showing you the upper growth with that really distinctive. You can't miss it when you walk into a nursery with all these container bays and you see that growth. It's really quite unusual. And then all of a sudden, you get in a midst of all that stunted witch's broom, dwarf foliage, you get one little branch that goes and is normal. It's really quite unusual and quite kind of curious. And this showing you the nickel sulfate again, and you can spray it or drench it. And I just have to be aware information is available if you want to apply this, and just make sure you use the proper quantities because you can, as I already indicated, create phytotoxicity with any kind of toxic over application of the nickel sulfate. All right, so another one now, tulip tree and magnolia scales. Now, these two scales are quite different than most of our soft scale species, and they are soft scales, okay? So they're phloem feeders, and they're gonna produce honeydew, and they're big scales. They're gonna produce a lot of honeydew. Now, instead of usually June and July, when most of our soft scales species are having their crawler periods, these are the exception to the rule. It's going to be August and September when they come out. And so your treatment's going to be a little bit different, and you have to start looking for them at different times. And there's only one generation a year, which is pretty much true with all soft scales. And by this time right now, certainly you can look at the growing degree days of the two scales that are listed there and what we presently are at. You can see that we are certainly having crawlers taking place now. These growing degree days are interesting. We talked about this during one of our meetings yesterday when we were preparing for this talk. And these growing degree day ranges can sometimes be a little bit confusing. Now, that doesn't indicate that, for example, with the tulip tree scale, it says like 2,000 to 2,600. That doesn't mean that the crawlers are only going to be occurring during that time. I think that range means is this is when you start looking for the crawlers and they can begin to occur during that range. Because certainly I've seen crawlers well past the 2,600 growing degree days. I've seen them in late September, and so we're well past, at least in central New Jersey, because we're already at 2,700 now, and the crawlers are still coming out. And so just be aware of that with these growing degree days. Remember also that these two scales, they only feed on woody tissue with all life cycle stages, and so they will not be on the leaves. And they do produce a lot of honeydew. And I have nurseries that have rhododendrons growing underneath a grove of tulip trees. And there's like 7 or 8 of them, big, large tulip trees. And this seems to be a perennial problem they've been experienced where they're infested with these tulip tree scales, all right? Because they're tulip trees, they can't be magnolia scales. I don't think I emphasize that enough. Magnolia scales will only feed on magnolia, whereas tulip trees can feed on both, either tulip tree as well as magnolia. That was an important information I forgot to mention. But with this particular example of the nursery, the magnolia, or rather the tulip trees, are loaded with the tulip tree scales, and they're dripping their honeydew down on the rhododendrons that they're field-grown rhododendrons. And they're big and they get this honeydew, and then the corresponding black sooty mold begins to grow in the foliage, and it creates a real problem for them, and they're really difficult to sell in that condition. And so typically they have to kind of clean off the foliage. I know soaps sometimes have been indicated as a potential solution, as just a small percentage of soap to scrape off the black sooty mold. And so that's something maybe you want to look at, or better yet, try to control the scales early. And those particular nurseries more than likely going to eventually start going with some systemics and apply them on the bark of the trees and have them take it up into the canopy and give it about a couple months or so, depending upon the material they use. And they're going to be using a neonicotinoid, and so that's what they're going to be forced to be doing. All right, so what do these things look like? And so this can be confusing as well, and I was confused by it. Kind of embarrassing. In fact, it was so embarrassing, I had a plant pathologist correct me. Boy, I just talked about humiliation, but that happened recently. And so there's the tulip tree scale there shown on the left, and they're kind of pretty good size. They're maybe up to about 1/4 inch in size, whereas the magnolia scales are bigger than that. They're maybe up to a 1/2 inch in size. In fact, the magnolia scales are the largest soft scale in North America. and they're quite large. And how do you distinguish between them? Well, you can look at the color. Usually the tulip tree scale has somewhat of a orange coloration to it, whereas the magnolia scale will be more of a light brown color. And also the major differences in their shape too. The tulip tree scale, if you look along the edges, it's kind of, it has an irregular look to it, that like an apron fringe where the it kind of flattens out a bit. Whereas if you look at the magnolia scale on the right, that just falls right down to the branch, just like right it's like a cliff right there. There's no fringe of an apron that's showing there. And so that's a good way to maybe distinguish. And this is when you have mature females. That magnolia there is showing a young adult female that produces initially that white powdery wax. It doesn't persist long. But when you see that, you know that's just a recently matured adult female magnolia scale. And so, all right. So this is, reason I didn't talk about, because I hadn't come to the slide yet, but just showing you the host plants on the tulip tree scale. It's tulip tree and magnolia, and occasionally some of these other ones. I've actually never seen them on those trees, but they're listed, but I definitely have seen them on tulip tree and magnolia as far as tulip tree scales. And another thing is that you're looking at the growing degree days, there is a bit of a difference. They're close, quite close, but apparently it's maybe about a 1 week difference. And so the tulip tree scale crawlers can potentially emerge about a week earlier than the magnolia scales. Not necessarily a big deal, but that's just something you need to be aware of, perhaps. And I've talked about most of that already. And so finally, they overwinter when they mature. Of course, the females lay eggs and they start crawling out as first instars, and then they feed for a while, but then they move to they only feed on the branches, and all life cycles are only on the stems and branches. And so 1 to 2 year old branches, you'll see these young nymphs, and they'll become second instar nymphs when they settle, and they kind of size up considerably. They kind of turn almost a black color, or at least a dark brown color. And you can see them on the 1 and 2 year old branches. And that would be your target for controls. And you can certainly use oils if you can reach them with oil sprays. All right, so it's showing you the crawlers there, the tulip tree scale. And this is, again, you can see the crawlers are quite tiny at first, dark color to them, light to dark brown coloration to them. I took this branch and shook them on that white piece of paper, and there's hundreds and hundreds of them can fall out very readily. They produce thousands of eggs, these large females, both the magnolia as well as the tulip tree scales. And there's the magnolia scale on the right. You can see the difference between them. This is maybe the one on the left's not so pronounced because they're all clumped together, but you see how the magnolia scale is kind of plumped up, taller. They're certainly bigger than the tulip tree scale, but and they don't have that apron or that flattened fringe that the tulip tree scales has. It's easy to get these two scales confused, and I've done it multiple times, I think, over the years. But anyway, just to have a key thing to remember, though, if the scale is on a tulip tree, it's not magnolia scale. It's got to be the tulip tree scale. Whereas with magnolia trees, it can be one or the other. So you don't have it quite so easy then. All right, and it's showing you the honeydew. They produce a great deal of honeydew, both scales. And this is your target. Now, these are the second instar nymphs on branch stems or twigs. And they're pretty easy to see, and you can view them with the naked eye. And so this is what you spray for if you try to control them with oils or soaps, because you can get control with both of those. But the thing is, when you have larger plants, maybe they're not going to be so easy to do. All right. All right, so I have to kind of finish up just for, you can read this on your own. There are neonicotinoids and systemics, acephates. They all work as a translaminar, although acephate really probably is not going to be a translaminar with branches. That's only going to be on the leaves and not be on the leaves. But you got the pyrethroids and carbaryl, they all work. But against the crawlers, that multiple oil applications or soaps may be required. And so this is my final statement with these two scales, is that you can wait you don't have to be in a rush to spray now. In fact, I wouldn't spray now at all because there's still a lot of, crawlers that are protected under the female cover, and she may even be dead, but the scales crawlers are still protected under there. And so it's been stated that you can wait until mid-October to treat the vulnerable second instar nymphs with any kind of an oil or a soap material if you can reach them. Okay, so that's a good thing to try to remember. All right, so I'm really kind of out of time. Well, what a surprise! I'm out of time. And there's the Douglas fir needle midge. Maybe I'll just show you some photographs so you know what they look like in the future. Um, they are, you look at the growing degree days, 200 to 400, and so they occur in May when the adults come out and they lay their eggs on the very young developing uh, needles, or even in the buds themselves before the needle have actually begin to expand. And they are maggots and they're fly larvae and they feed within the needles. You can see the distortion that they make there. Okay. Let's move through this, and you can see one of the needles up close where you can see the dark color in the maggot, and there's at least two of them in there. You see that distortion and a really kind of a swollen area. It makes the plant look really unappealing, and so they can begin to be seen. Let's go to the next slide, and kind of going clockwise real fast. There's the adult in mid-spring, maybe May, when they come out from underneath the soil. They drop to the soil in late fall as larvae, and then they pupate in the soil, and then they emerge as adults in the spring when the soil temperatures begin to rise. And there's this white circle of an adult laying egg in the bud of a Douglas fir. And by the way, these only feed in Douglas firs, no other plants, and they're really most commonly seen in the Christmas tree farm industry. Okay, you don't see these too often in the landscape. Early summer is kind of a little bit of a discoloration taking place. You get mid to late summer now, you begin to see much more pronounced distortion, discoloration. The leaves are starting to darken up. By mid-fall, they almost become almost a dark purple coloration, and they really become pronounced. Something that looks really unappealing if you're trying to sell a Christmas tree. And then the late fall, maybe November, as late as November, they finally fall out of the leaf, they drop to the ground, and that's where they pupate. As far as controls, I really have to stop, so I can't go through this. One thing is that you can use a trap and you can put it underneath the tree. Remember, they overwinter underneath the tree they fed upon, and they pupated there, and they emerge as adults in May. And so if you want to try to determine when the adults are going to be present, even though they're going to probably be active in May, you can use that as a guideline. But even more precise, you can use a trap that's shown in the photograph. And there's a box with maybe you have a wax coating around it, protected against rain, or perhaps a plastic type of a box of some kind. But you need to cut a hole in it and put a glass jar in there. And so when the adults emerge, it's going to be dark, but they move to where the glass is inserted in the side of the box. And they'll move into there and they don't go away. And they just want to be attracted to the light, and they die in there. And so you can see when they become active. And so what you do is you really try to spray against the adults. Apparently the systemics against the maggots themselves that are inside the needles are apparently not all that effective. And so you got things like pyrethroids primarily, or organophosphates, as sprays against the adults. And so you're spraying the trees, and so when the adults begin to attempt to lay their eggs, they're killed by the protective sprays that are there. So, all right, I think I'm out of time, and so let me turn it over with plenty of time he has, which is Dr. Tim Waller.
All right, well, thank you very much, Steve. Hey, everyone. So we're going to be talking about our good friends, snails and slugs. There's a lot of information here. Snails and slugs are always a problem in situations where we have a lot of moisture. That is exacerbated in situations where we have shade. And it's further exacerbated when we have moisture, shade, and our plants are in too high of a density. We've all seen snails and slugs moving around in the nursery, whether that's in high tunnels or in situations where maybe the weeds got away from us. So these are not insects. These are mollusks or gastropods. And they take a little bit of a different approach when it comes to controlling them. There are differences between slugs and snails, but what's important to note, though, is that once these populations get established, particularly in situations when we're holding on to plants for multiple seasons, we can have some really explosive population buildup. Uh, these organisms have to be able to get back to moisture. They like feeding at night. So if we start thinking about it in through that vein, it gives us some ideas of how we can control them through cultural practices, as well as in our chemical practices, which we're going to talk about more in a second. So we probably have all seen the type of damages caused by these mollusks before, these gastropods. And the reason for that is because of the way their mouth parts are shaped. They have a rasping type feeding behavior, this giant saw-looking thing, their radula that is going through. And oftentimes we'll see damages in kind of these linear fashions in much more succulent leaf tissue. If the situation is right, they will go for a little bit of a stockier leaf, but generally we're going to see this in more of our more succulent leaf tissues. One of the big things you can do, if you have snail issues, you're probably going to see dead or desiccated snail shells, but it's always a good idea to keep an eye out for the movement of their slime trails. Kind of gives you an idea of where they might be, where you might be able to locate them, where you might be able to treat for them. So there's a couple things here that I wanted to point out with this. So there are some materials that are made for the mollusks, and I'll start here with the metaaldehyde, which often has the addition of a carbamate in the form of methoxycar. These materials are definitely older materials. There's some differences in labeling. They have been used for a very long time. Some populations are able to, there's some level of resistance that's being built up by the localized communities. The kicker with these, though, is that they are dangerous to other animals. So being cognizant of where your product is going out into, is it going into the landscape? Is it going to a garden center that is very animal friendly, etc.? Those are things to keep in mind. And the way that these work is they're more or less a bait and kill situation. And they stop the moth's ability to maintain its moisture, and then they desiccate. Again, remembering moisture is so incredibly important when it comes to these particular pests. Then we have iron phosphates. These are newer materials. It works in a similar kind of way, at least on the surface, as BT does, is that once the snails have consumed this, it stops them from being able to metabolize things or being hungry, quote unquote, and it shuts them down. So it doesn't kill them immediately. It takes a little bit of time, but these materials are much safer for a lot of other organisms that are out in the ecosystems. So when it comes to our cultural practices, for those of us that are on the line that have some interactions with the landscape, it's very important to note that of the natural predators, particularly of snails, are fireflies. Their larvae feed very heavily on snail larvae. So if you're experiencing this issue in the landscape, be mindful that it could have a really big impact on one of our favorite insects that's out in the environment. But there's lots of other things that eat them in a naturalized system is very important for the food web. Like I said before, overhead irrigation is a big deal. We want to avoid anything that is going to trap humidity, spacing our plants out a little bit more. Basically, all of the things that would be good for disease prevention are also really going to help us out when it comes to controlling our snails and our slugs. There's different ways that you can trap for them or at least monitor for them. These can be highly effective in kind of getting a baseline of what you're up against. So just remember, like with a lot of our diseases, the drier we can have our production, the better off that's going to be when it comes to controlling a lot of these moisture-loving organisms. All right, so I'm going to talk about kind of the late season boar, clearwing boar. And this is the banded ash clearwing boar. Unfortunately for us, throughout New Jersey, ash trees are something that we are seeing in lower and lower production. They're being decimated out in the natural areas due to emerald ash borer, which is a bupressid. That's a beetle. But this one is a much later clearwing borer than all the others that we've been talking about throughout this season. Um, and their main host plant are the ashes. There's a lot of good information here, and I feel free to look at that as well. So like with our clearwing borers, they're going to have a circular to an oval shape exit hole. Sometimes you'll be able to see where the clearwing is pupated out, and out from the side of the bark came the clearwing borer in its flying adult phase. And this is fundamentally different than something like emerald ash borer or bronze birch borer, which are going to have characteristic D-shaped holes because of their larval stages and how they ultimately mature. We just wanted to bring this up for completeness sake. The trees that are shown here these are green ash trees. They were not killed by emerald ash borer. Rather, they were taken out by this banded ash clearwing moth borer. So just remember the exit holes are really important when it comes to figuring out what you're up against. As with all of the other clearwing borers, pyrethroids that are going to give us some level of extended protection are really going to be the most important contact materials. There's some indication that systemics applied directly at this time point where eggs are going to be beginning to hatch, as well as some biorationals, can be effective. However, in a production system, the pyrethroids are going to be really valuable to give us that kind of extended protection. This is also a great use for our pheromone traps that are going to call in males and give us an idea of when these pests are around so we can properly control for them. All right, so thank you, Steve, for 8 minutes to go through a very complicated fungal issue. And I wanted to talk about verticillium wilt. Verticillium wilt is a soil-borne disease. There are a number of species that are of concern, depending if we're looking at woody ornamentals or we're looking at more herbaceous plants. But this can be a really big deal problem that I know we've all dealt with in one way or another, whether it's in the nursery or in the landscape. I'm going to show a life cycle here in a little bit, but what I wanted you to see in this image here is oftentimes we're going to see damages that are in that blackish to greenish coloration right at the interface between the xylem and the phloem. This pathogen can be harbored in a gigantic list of weed species where it can continue to be persistent in a very virulent form. They also produce these structures that enable it to persist for very long periods underground. A lot of times we start to see these issues very early in the year or very late in the year, depending on drought, stress, or status, because they're no longer able to function correctly. This is one of those pathogens that it's very important to consider working with a diagnostic laboratory. So I pulled this image up here, verticillium wilt. Oftentimes you're going to see flagging or parts of the plant that are starting to die first. This could be at the apices of the plant. This could also be in individual branches. You also have instances, particularly in nursery production, when there's a high pathogen load where we're going to have full plant decline in a very rapid situation. So verticillium wilt symptomatically can take on a lot of different forms. And I didn't note it on the last slide, but red buds and maples are often, verticillium can go after hundreds of plant species, but maples and red buds are oftentimes where I see it in the commercial side of things. So this is a dramatized life cycle of verticillium wilt. And what I want to draw everyone's attention to is that in that white box there, if we're starting on the right-hand side, this pathogen creates microsclerotia, which means that basically we could remove it completely, but if we do not get all the plant debris, we have not fully gotten rid of this pathogen. Creates these survival structures that ultimately, once they come into contact with root tips, initiate the infection again. They get into the roots, they start to work their way up into the xylem tissues, and we're having a complete disruption of the plant's ability to shuttle nutrients, to shuttle water around. And ultimately, it leads to partial or complete plant death, meaning that it is eventually moving towards complete plant death. So unfortunately, there aren't, you may see verticillium listed on chemical controls. However, there's no highly effective treatments. There is fumigation that can be effective, but when we're talking in a nursery situation, it may not be legal nor practical to go in with that type of approach. And you really need to get an appropriate diagnosis. If you're dealing with something like Phytophthora versus verticillium versus vascular streak disease, these are all different approaches that we're going to be able to have to take. It's a really good idea to rogue symptomatic plants once you suspect verticillium is at play. And when you have symptomatic branches, and I alluded to this earlier, in nursery and greenhouses, if we have symptomatic branches, you've shown that it is verticillium, you are not going to be able to save that plant, and you need to get that out of the system. If you're in a landscape situation, you can remove symptomatic tissues, but noting that this pathogen is colonizing the crown area of the xylem, you're ultimately going to lose that plant. And sanitation is so critically important when it comes to working with this pathogen. And particularly, we grow a lot of maples, we grow a lot of red buds in pot and pot production, as well as in-field situations. So I just want to point out to everyone that if we have plants that are succumbing to verticillium wilt in a pot and pot or in a field system, it's really important to evaluate how clean or how sanitized your socket pots really are. This means changing out emitters. This means going in with something like quaternary ammonium and sanitizing the sockets the best we can. Just thinking about where this pathogen could be coming in. And this is just another plug for this. So verticillium wilt, like I mentioned before, a lot of times we're going to see that ring, and oftentimes it's at the interface between the xylem and the phloem. A lot of times it is very, very consistent where you can go in with a knife and you can pull it apart, and you'll start to see the streaking of these blackish browns to greens in some cases. This is the reason that it's so important to get a diagnostic look at this is that we also have this issue that's becoming more and more prominent. Although its occurrence in New Jersey is not quite understood just yet, but we also have vascular streak dieback caused by a completely different pathogen, and there's a lot of back and forth about what pathogen is actually causing it. However, knowing this helps arm ourselves to be able to deal with the problem at hand, and spending less time guessing about what we're doing. So the diagnostic approach is really important, and you need to have someone take a look at these plants. All right, so I did it with 2 minutes to spare here. We really appreciate everybody coming in. Some of the slides that you saw today, or some of the information, were people sharing information with us being able to work through that information and be able to share with everyone else. We are always available if you want us to work on something or you want us to bring something that we could share with everybody. We really appreciate that type of collaboration, and we're here for everybody. So next time we are going to be talking about ticks and some of the science behind it and things that are going on in New Jersey. So you might be asking yourself, why are we talking about that in a nursery IPM session? Well, we all work in agriculture. We all work outside. We all know people who have dealt with tick-borne diseases, and being aware of these things are really important for us. We're all managing people or working with other people. So having an idea of what we are up against is something that's really important to all of us in these agriculture industries. So with that, thank you very much for attending. We're going to stop the recording now.